Effect prediction of sequence variants. So first let's go back to what effects mean on the level of DNA or RNA. There are substitutions, so single nucleotide changes, A to C for instance. There are insertions, which is essentially addition or removal of one or more nucleotides. Indels is just longer insertions and deletions. There are duplications, which essentially are insertions too, just of the same region. And then there are inversions, so essentially you flip the DNA around so that it's read the other way around. Now, on the level of proteins, the same thing happens. So there are substitutions, single substitutions. Now, these are sort of nonsense versus non-synonymous. So only the non-synonymous change the amino acid sequence. We would refer to them as SAF. But then there are also insertions, deletions, and duplications. The same story. Inversions lead to, obviously, to different sequences, or tend to lead to different sequences. No, I believe there must be some that don't lead to different sequences, but most of them lead to different sequences. Now, on the level of words, today these single amino acid or the single nucleotide residues changes are called SNPs, single nucleotide variants. There historically was this issue of polymorphisms. Does anybody know why they existed or what that meant? The actual word that you all may remember is SNP. The word SNP by now is historical. When you started studying, everybody talked about a SNP. SNP is no longer relevant. Do you know what the idea behind the polymorphism is? The idea is ultimately that you observe it more than a certain frequency, and the reason for having that is because in doing the sequencing, if you did not observe it more often than some number, you would believe that was a sequencing mistake. Okay? So this is simply the word changed. Historically, SNP meant you have seen the variant in more than 1% of the population. And that is completely off because sequencing now is good enough. If you see one variant, you see the variant essentially 10 times genome sequencing or something like that. You believe this SNP is existing, right? That's why these words have changed. So then there is a mutation that is caused by something on the level of the amino acid. Again, I talked about the single amino acid variant. And then there's sort of this word of 19 non-native. Essentially, on every single amino acid, you have a native one, and then you have the 19 possible alternatives. Okay? That is what I refer to as 19 non-native. Now, there are some SAF possible variants. SAF possible means essentially you have you essentially reach it by a single SNF. So you change a single nucleotide, UG, the coding for a W. You could change it to an F, but changing it to an F, you need to change 2 nucleotides. So you would call that change of the W to the F as SAF impossible. So this is not reachable, or SNF impossible. I'm sorry, I should change this. It's not SAF possible, it's SNF possible. So it's an amino acid change that comes about by a single nucleotide variant. This one here would be UG to CG. You simply switch the central G to a C and you get a new residue. So W to S is a SNF possible change. Okay, and this plays a role because some prediction methods actually use this feature. In order to see whether we can differentiate the effect that we would reach in the molecular experiment. So this implies that in nature you cannot get 19 non-native by single SNFs. Some variants cannot be reached by a single nucleotide variant. Again, we have enough space in evolution to have more than one change, so you can change every single amino acid. Could in principle change every single amino acid. But again, so in an experimental setting for proteins, we can do things that are not easily done on the level of the genome. Sickle cell anemia is one example where we single glued to valine. By the way, this is just one of those small details. Some of you may remember that on a previous slide, I called this E6 to V. If you observe a little bit more carefully, what this one here says glue, which is E7 to V, right? It's not a typo. What is it? Any idea? It's just one of those quirks of molecular biology, yes?
Yeah, one of them counted the methionine.
Exactly. That's exactly the point. How do you know?
Talked about it.
Are we when?
In one of the lectures. I don't remember.
At least, now that I'm impressed by you, I feel a little bit better because all the others are with me on this one and I've forgotten about that. So I have all the others. Wow, I'm impressed. So yes, the historical, the real first letter is a methionine, but in the biochemical experiment you have always cut it off. So the sequences in Swiss prot often come without the methionine. And that is this is one of those small things I wanted to make you aware of, because it's just one of those simple things you can stumble over when you do sequence analysis. Anyway, so this particular variant here leads to a different aggregation of cells, blood cells, and that leads to a bad disease. Now, synonymous SNFs, so nucleate variants, that in fact change the nucleotide but not the amino acid, so these are in fact the synonymous ones, how can they have an effect? Can you imagine that? So first of all, not many of these are known, so it's not one of those things. All synonymous SNFs will have equally probability of having an effect as one that changes the amino acid sequence. Not at all, but some of them can have an effect. Can you imagine the mechanism by which that happens? Yeah?
Maybe if the coding changes, I think it's not exactly...
Yeah, but wait a minute, the coding is not changing.
No, I mean if the tRNAs change.
Yes, so immediately, so we have a regulatory issue. That would be one way of doing it. So here we have one change in the fibroblast growth factor, which ultimately is the way the translation speed is done, and that ultimately is the issue, right? So we express faster, shorter. That can have an impact on, or that can cause a disease. Cystic fibrosis is an example for, again, a single effect, a single variant effect. There are many more effects. There's copy number variations, for instance, in Huntington, where you have this, if you have a certain number of Qs is okay, if you have more than that you get a disease. Then there are repeats. Let's not get into all of these. What we are going to focus on really is single amino acid variants, so the SNFs that change the amino acid. And the first generation methods, as we already briefly discussed, the simplest idea here is sift, where we essentially say that if it is in aligning, if that variant is similar or is in the blossom matrix something that is accepted, we take it as neutral. If it is not accepted, we take it as non-neutral. And that's essentially why this sift is called sorting intolerant from tolerant. So not accepted, effect, tolerant, neutral. Okay, that's the idea. The people behind it are Stephen Hanikoff from Blossom Matrix, and Pauline Nick, who is now in Singapore, in Astor. And again, so this is an old slide. Since she's young, old slides make a big difference. But very early on, with this paper and the follow-up paper, she had a big success. And again, the idea is, I already said it, change in biophysical features. And in setting up that method, all you need to do, essentially look at the Blossom matrix, define a threshold when you say more or less, end. And then you establish how well it works. They established how well it works for 3 proteins for which they had about 6,000 mutants in 2003. And that was the first method that was available. So Polyphen was the next, and Polyphen, here you already sort of polymorphism phenotyping. You already see those were the days of SNPs, so this is still when we have polymorphisms, but it's the same thing, so SNPs. The main person behind it is Shamil Sonayev, now at Harvard Medical School, who again has increased his influence by far more. And the idea now, as opposed to Blossom, we're not asking just what is the biophysical effect, but what is the effect at the background of the alignment. So if I have this E or E7 to V, so it's counter methionine, if I have E7 to V, so the question is, at that position 7, do I at all see a V? Do I, for instance, see 60% of the time an E and 40% of the time a V? If so, I would say this is a neutral variant. If I never saw that, if I sort of saw at position 7 only E's, or something such as E, 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 Q, 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 or Q, Q, then I would say the variant to a V has never been observed in that family, so it is unlikely. Now, what would you do if at that position you saw so you have 5 proteins aligned in that family at position 7, and those are the 5 residues you see. Now you do not see a V, okay? That's the way I want to have it. What would you say? Is the V accepted at that position or not? Or would you argue that so, would you say that this is less here than there? So obviously there is no V in any of the two, right? But if your prediction for the top example here were that this one is, the V is an effect if the alignment looks like this, and the V is an effect if the alignment looks like this, is any of the two stronger for you? Why?
Top.
The one on top.
No, the one on top is not conserved at all.
Yeah.
So it's more likely that the V won't be accepted.
That it won't be accepted or will be accepted?
Will be accepted.
Mm-hmm. Can you say something? So yes, I totally agree with that. Can somebody put something on top of it? Looking a little bit at the biochemistry of the story. Finally, the leucine and the isoleucine look a little bit more like a valine. So they're hydrophobic residues, right? So you're not seeing a V, but at least you see sort of residues that are compatible with that feature of hydrophobicity at least, okay? So that would make it even in some sense more neutral, right? But you're totally right. So this is most likely a prediction of not accepted, so effect. And this most likely would be a prediction of neutral. And again, the prediction of neutral would be strengthened by the fact that you have at least in 20% of 40% of the cases some similarity of the effect, the hydrophobic effect. Okay, and that is the same idea. Now we get into SNAP S3D, and SNAP S3D is again a similar story, but now the argument that comes from the group of John Mould here, and again, just look at the numbers here. This, in fact, was even published before SIFT, I have to point out. This one was published after SIFT and Polyphen. And the idea is now, we still use the alignment, but the point is, if I know the protein three-dimension structure, I can distinguish between residues that are on the outside and proteins that are in the residues that are in the inside, and different things will have different effects on these two. Okay? So inside of a protein, what type of variant will have an effect? And outside, what type of variant will have an effect? So if I measured a bunch of SAFs and I know they have an effect, and I look at the half that I then can map to the surface and the half that I can map to the inside. Yeah?
Whether they are hydrophobic or not.
So you mean that there are variants inside that are hydrophobic to not hydrophobic? Is that what you're saying, or?
Yeah, the inside it should be hydrophobic and on the outside hydrophilic.
Right. So you will. What will you more see on the outside? What will you see more on the inside? So you will see, essentially what you're saying now is that on the outside you will see more variants of hydrophilic to hydrophobic, and on the inside you will see more hydrophobic to hydrophilic. Is that your statement?
That changed the structure, yeah.
True. That, in fact, is the one I was hunting for, Daniel. The second one. Yes, that's true, but the second statement is what I would call the more important one, whether hydrophobic or not. So what you will see inside more is that you mess up function by messing up folding or messing up something in the structure. Okay? The function now changes, and what's the opposite way of looking at it from the surface? What would you see there more that is measured as an effect? It's difficult for me to ask a question. I make it so convoluted so that you not even really know the answer. When I say convoluted, you don't know what I'm asking. I'm not good at asking questions. I'm sorry for that. So what I meant is on the surface, ultimately, if you see a variant on the surface that has an effect, what do you immediately suspect what that means? So you know not much about the protein. What you do is the following experiment. You know that the protein is bound to something. Okay? What you can measure is the delta G of binding. Okay? You don't know anything about the three-dimensional structure. You don't know where the binding site is. You don't know anything of that. You know they bind, and you know how strongly they bind. Now you put variants, and you have a set of variants that mess up this binding. You happen to have a solvent accessibility prediction. And for some that mess up in action between these two, they're inside. And we would argue that they sort of mess up structure in some way. And for some they mess up binding and they're on the outside. What do you believe what they do?
They are probably on the binding side.
Exactly, Navi. That's exactly the point. So the underlying model is when you mess up on the inside, you mess up the structure. And sort of the thing does not bind, not because the binding side is affected, because simply it doesn't bind. Okay, it doesn't fold. It doesn't do the right thing. While when you mess up something on the surface, or when you have a variant on the surface and you measure an effect, then you assume that this is because that essentially is in the binding site, or determines the binding site in some indirect way, by coupling, by connections, by residue-residue interaction. Now, what all of these methods have in common here is that you essentially have no parameters to fit. You have a threshold for what is blossom effect versus not effect. You have a threshold for how do you compile alignment conservation, and then disagreement with what you see in the family is a simple score, and then you again define more or less, right? As simple as that, as a simple threshold. Here you do something like that, you just distinguish between inside and outside, you distinguish between some sort of contact preferences that you know, but ultimately again, you have very few parameters you optimize. When you want to do machine learning, you need data, okay? And that begins to get into the point where you have to collect data. When you collect data, you face the problem that some of the data is on the level of an organism. You see variants that affect the way the organism looks, okay? Some of the variants are on the level here, the other, let's call it the other extreme, where you have a detailed molecular biochemical change. The delta G of binding of two molecules, and you have an effect of that variant on that delta G. In between, I'm calling that in between, so here the idea is you see a change of the electrostatic potential. You're not necessarily measuring, but you see that there's a change, and you know that the electrostatic potential sort of attracts the DNA in this particular case. And if you change that, it will not attract DNA. You don't need to make a measurement, okay? You just have to see that. In this particular case, the idea behind that figure is essentially you have some variant, and that variant has infuses a color change or something like that you can see, right? And again, when you see a color change, there's this judgment call from an experimentator to say color change, yes or no, and something in between, right? And these are very, very different scales of measurements. And now when we collect all of these different scales, collect all of these different data, and we collect it for all organisms for which we have anything, then we get to a data set that actually mostly is determined by a database from Nishikawa, the protein mutant database, Kawabata and Nishikawa, and mostly essentially uses the delta G or delta G binding here. And when we collect all of that, what we get is about 40,000 effect and about 14,000 neutral. So about a fourth neutral, okay? So when you now do a machine learning on that, so the first issue that you have is a sort of not that big data set, but it's 40,000. It's a good number. To start with, the first issue that you have is with the imbalance. How can you correct for the imbalance? Other methods to address this issue that essentially would sort of simulate data, SMODE is one of those examples, where you essentially take the negatives and try to in the computer simulate similar data. So for instance, in a protein, you could use similar proteins and sort of map the same variant into a protein. That would be one way of doing it. In the protein world, it's more typically, or in our experience, often doesn't work. At least if done correctly. Anyway, so we can balance that. Another way of doing that is to sort of introduce not quite in silico variants, but use the following idea of enzymes. So let's assume we have two proteins, here and here. For both of those two proteins, I know that they are enzymes, and I know the precise enzymatic activity by experiment. So I have two experiments telling me the same hydrolase in the detail of the hydrolase. Okay? Now I simply can look at where do they vary, and I say wherever they vary, they are neutral. But I cannot do that if they are 30% sequence identical, because then there will be long regions where they could do something completely different. If I had a long region, they could bind DNA in that region. But if these two are similar enough in sequence, say 90% sequence identity, then whatever they change in, and the 10% where they vary, since they have experimentally measured the same interaction or the same reaction, then I can assume that whatever else they have, the few residues in between, they will be neutral. Okay? That created in our particular data set a total of sort of 41,000 neutrals, so effect and neutral now is sort of on equal grounds, numerically at least. Not quite on equal grounds, because in some sense there are more mistakes in these than in these, and in silico neutrals. And then we can slice this by solvent accessibility. So buried versus exposed, effect versus neutral, and you see that the exposed ones, there are fewer of those here. You have more buried that have an effect. Let's just say ratio neutral to effect is higher, so there are more effect in the core than on the surface. So the inversion, or there are more neutral, there are fewer neutral in the inside than in the outside. Why is that? All the function is somehow on the outside, no?
But there are also many loops that doesn't really need to be conserved.
So ultimately the idea, again, imagine a cell to be a solid state, it's immensely crowded in a cell. Still, most of the protein surface is not binding to something else. And this ultimately is what the background here is. Most of the surface of a protein is in fact not directly involved in binding, is somehow, and that means it's somehow free to change in some sense, right? That is what comes out of it. Anyway, maybe you see it best here in these pie charts. Neutral is green, effect is red. You see buried and exposed is very different. Okay, now we can predict. We can develop prediction method. I call it also second generation because now we have enough data for machine learning. There is a third generation that uses sort of much larger data set, that looks at diversity, that looks at disease data and all kinds of other things. And I will not really get into that. But the first application of SNAP that originally was done by Jana Bromberg, again, who is now a professor at Rutgers University. But before I get into SNAP, let's just give a few other methods. The first one here is from the lab of Tom Blundell, and essentially that is the idea to go from wild type to mutant through the thermodynamic cycle and to measure the effect, the delta delta G versus the calculated delta delta G from energy models, and that ultimately gives you one way to sort of recapture the experimental data by an energy potential here. Straight line means you capture it, and straight line essentially means you predict the effect of a variant for the binding on the energy level model. Okay, that is one way. So essentially you you're not machine learning to predict effect of binding directly. You are building a model that explains the biophysics of binding that fits the experimental data of delta G measurements. Okay? Here is an idea from the lab of Marianne Ruhmann. And that, in fact, looks at the changes in binding energy from 3D structures. So you look at the three-dimensional structure, you compute the binding energy change, and essentially, you like pop music? Yes, so is that what it is?
Not about the name.
So she has a bunch of tools, and so pop music is only one of those. Marianne has a good way of choosing really names that you feel like that makes you feel good. Or at least it has the same effect on me that it had on you. Anyway, so you look at the change in the binding energy that is computed from 3D structures, and ultimately, first they tried it as a model that sort of simulates MD calculations, and then they went into machine learning with this. So initially statistical potential. Now we get into something done by Rita Casadio and Emilio Capriotti, Piero Farizelli in Bologna, iMutant. So again, now we're looking at experimental delta G measurements and predicted, so essentially a machine learning from the sequence environment, what would be in that particular case the change, the effect of that change. The delta G learned from a database called Protherm. Josef Weissmann has done something similar initially with a model, so they went directly into machine learning when they first published that. That is the first publication, 2005. He had something before this one, so this one is already a machine learning application, sort of the next level, where again, this ultimately is how does a particular sequence variant change the binding energy, which in fact also means how does it affect function, one particular aspect of function. Provian is a method that essentially is based on the sift idea, as an extension of the sift idea. And the extension of the sift idea ultimately is you compute effect on subfamilies. So the Polyphen would build a big cluster, and now you look at subclusters and you ask again, in these subclusters, does it have an effect? And that gets a mutation taster is something that is sort of is similar to that what I had before. No, this is actually something that does OMIM based and is more human focused and essentially learns to map OMIM type of variants, so OMIM, online mammalian inheritance in man, Mendelian inheritance in man. And it's a database for single amino acid variants that cause disease. Okay? And they want to distinguish between the variants that cause disease and all other variants. And ultimately the way they do it, they argue when I do an alignment to primates and species that are relatively related to human, and I do see that variant, then it's neutral. If not, it causes an effect. That's mutation taster. So this is again, if you want something like Polyphen with different ways of doing the alignment and a machine learning device underneath. Here's an evolutionary action. That's the Olivier Lich-Charge. So this is essentially, and I'm not sure why I didn't write the formula. Ultimately, there's a method underneath which is called the evolutionary trace method. The evolutionary trace method looks at trees. So this is my protein, this is a related protein, there's another protein, there's a tree, and in this tree there are sort of residues that are important to determine whether you go into that or that direction. And these are typically particular variants on the surface, okay? Tree determining residues is what people call that, okay? They determine whether you go to that specificity or that specificity, okay? And now the idea here is build this tree, find those residues, and compute the effect on these residues in terms of variation. And then ultimately he phrased all of that in an equation that is derived from these trees, computes a similarity or an evolutionary gradient for these trees, evolutionary gradient that then is a gradient for every single amino acid. So ultimately you then in the alignment have a gradient, and you can apply the gradient and you can predict the effect. in that sense. It's not directly machine learning, so you fit some data, but it's not on the scale of thousands of data points. CAD, and I don't have a photo here, is Martin Kirchler, who's now at the Charité in Berlin. So it was originally published in Schender's lab in Seattle, the city under Vancouver. I got it. Washington University. So essentially, the idea behind that one here is we train on variants without effect that differ between human and human chimp, common ancestor, and appear in large number in the human population. Essentially, it takes apart things that we see in evolution in relatively similar organisms and things that cause disease. These two things are taken apart, and CAD is one of the best existing methods to predict it on the genome level. So CAD is a method that essentially works, unlike whatever I showed you so far. CAD can predict effects on on SNFs that do not change amino acid sequence, right? Because the alignments underneath you can do as well for the non-coding part. That's one of the many strengths of CAD. Here we have another idea that comes ultimately from Boris Reva and Chris Under. It is somehow similar to the tree, which may be bad that I put it before, because historically this was earlier, where again, if you have a family alignment, in that family alignment you see that 4 have an E, 3 have an N. Okay, so far, for what Polyphen said, and what Provian is slightly different, and evolutionary trace method is different, but for most of what I showed you so far, what essentially the highest probability would be the E, the second highest would be the N, right? So if I assessed how likely is it that if I have a variant, if I'm in that part of the tree, and I say N to E, right? Then for the methods that I discussed so far, they would all say, well, anyway, more than 50%, so 4 out of 7 are E's, 3 out of 7 are N's, so N to E clearly is an acceptable at that position. While this method now would say, well, wait a minute, if you're in that part of the family, you have 100% N's there. So changing that to an E, although at that particular position here is relevant, you actually do something that for the species you are is not good. Because this, in fact, is a specificity determining a residue that determines how you react to certain things. It's not the binding site, but how you react to something. And this, in fact, means that you change that. In the sequence that you have, this will have a strong effect. Okay, that's the philosophy underneath. Mutation assessor is available as a method as a web server. Now, there's another set of methods that goes back to work from Deborah Marks, and actually also Chris Under, evolutionary coupling. So we have 2 columns, and ultimately this is also 2 columns, and we see that 2 columns have some correlated, they are evolutionarily coupled, meaning that I have some coordinated change between here and here. This could be the other way around what I saw before. Say we had 3 up here, 4 down here. Say this is large, this is small. And this is small, this is large. So it's a flip movement, right? So you mutate something from large to small and from small to large. And the assumption is that you had to mutate in order to accommodate the same binding site, which means that you believe these 2 things are close in space. Okay? Idea is clear. Evolutionary couplings, right? Now, close in space means then you switch that around, and switching that around, essentially you look at correlations on the level of evolutionary couplings, on the level of the alignment, and from these correlations you try to predict correlations in 3D space, you try to predict contacts. So that is one way in which you can use that. Then you can actually use that to predict whether you can predict the structure with that. You can predict protein-protein interactions. But you can also assess how a variant is going to affect the structure, the protein interaction. It's going to affect function, right? Not only that, you can also predict the effect of more than one variant. So in this particular case, so we're still talking about SAF, but two SAFs. You can look at, if I had these two variants that are coupled here, maybe the change from large to small is bad, from small to large is bad, but if I see them both at the same time, they compensate each other. And in principle, the method could predict that. In fact, so there comes a prediction method out of it, and the method here, and I have to update the citations. It was his PhD thesis at some point, has been published since. So this is a method with which you can predict variant effects, and for many of the experiments for which we have high throughput variation, in the sense that for every single position you have you test 10 of the 19 possible variants. This is the best method, at least at the time it was published, the best method to sort of fit those data sets. The problem with that method is you need huge alignments, and this ultimately has to do with, in order to distinguish between evolutionary couplings that are meaningful of connections or contacts and those that are not, you need huge alignments. Huge alignments in this particular case means thousands of family members. In fact, the method gets better if you have 50,000 than when you have 10,000. So really big families. How many big families do we have? I said 50,000. Is there any family that has 50? Obviously there must be one. Otherwise I wouldn't say that. Do you believe there are many? What does that mean? No, so Daniel says not, but what does it mean not? Can you formulate not as a number? No. If I told you there are 5 that have more than 10,000. Are there more than 5? Everybody will be happy with more than 5. Would everybody be happy with more than 500? And that is sort of where we are roughly. I believe your no is most likely right. I'm not entirely sure at the moment. In the past it was certainly right. I'm not sure it's still right. But that is what the ballpark we are getting. So a few years ago, Christina Rengo made this observation where the 300 largest families accounted for something in the ballpark of 40% of all the sequences. 300 families, 40% of all the sequences, okay? That gives you some idea how immensely huge these largest families are. But actually, again, as you sort of correctly suggested in your no, there are not many of those. I'm going to get over this one here and look at a few ideas of the SNAP from Jana Bomberg through machine learning. So again, we are going to have the situation where we have 13 adjacent residues coded for in the same way that we did for interaction, protein secondary structure prediction last semester, or for interaction binding. Now the output is effect versus non-effect, or versus neutral, there's animation again. The types of features are what your typical suspects. Conservation, biophysical features, part of motifs and all kinds of things. But now comes an important new feature, which is a delta feature. So I'm not only going to put in that the particular window or the particular residue in the variant is predicted to be in a helix. I'm also going to put in whether that particular variant that I try to predict for, the E to V for instance, whether that changes the preference for a helix. Okay? And I'm going to do that for other input features too. It's not true that I only use this delta feature, because that was not the best. So sometimes, we have a mixture of these delta features, so change by putting that sequence in on that feature, change, so the delta feature doesn't work here, but the delta feature is for biophysical features, for solvent stability, secondary structure, residue flexibility, and all of delta again means I put in the other residue, the variant, the SAV, and I predict again secondary structure and I ask, are these different? And that's the delta, right? And in many cases we use delta and original feature, and then we also use somehow annotations. As always, there is sort of a score. In this particular case, we are going to plot data such that minus 100 means strongly predicted to be neutral, plus 100 means strongly predicted effect. Let's begin with the overall performance. First thing, we're looking at a data set that is in fact has not been used for training. And so when we initially published that in 2007, there were not that many methods available. Again, today I assume there are over 200 available. And those days, for full proteins, we had a lot of variance. And one way of looking at this, so this the overall two-state accuracy, is that ultimately this method that we developed sort of did slightly, at least using annotations, even not using annotations, somehow on average, for most of the cases that we looked at, did slightly better than what existed. Okay? So in all the columns, not in all columns, for HIV protease, for instance, sift was better than our method. And in those days, that essentially was what it was. Then we asked, how good is performance for different states, buried, exposed, buried, red, exposed, blue, for the effect, for the neutral. For effect, the best prediction is for buried. For neutral is the other way around. Then there is, of course, this issue about the reliability index. So both of these here are predicted to be effect. This is an effect, this is an effect, but this effect here is predicted much stronger because the difference is much stronger. The difference is increasing in that direction. What this axis shows is cumulative percentage of residues for which I predict, and this is the accuracy. Now what I mean is, if I go in my curve to the left, I'm not going to consider this case. I'm not going to evaluate it at all. I'm going to say my threshold must be higher than a difference, say, of 0.2. So I will not count this as incorrect or correct. I will just not count it. Okay? I will just completely ignore it. So I will essentially, answer on the left is only stronger than that, more than some difference. And the strength is not on an axis. The strength is actually the number. That's the reliability index here that is defined as an integer between 0 and 9. 9 is very strong, 0 is very low. Essentially 0 means as long as this number here is higher than this one, I make a decision. That is what the 0 stands for here. When I look at the 0, I make predictions for every single residue. So as long as I say whenever one unit, the unit that is larger is the prediction, I will always find one unit that is larger. So I will always make a prediction. If I do that, then my average accuracy for the effect variance is in the ballpark of 77%. If I allow myself to choose the sort of 50% strongest predictions, which are in fact the ones with a reliability index higher than 4 or something like that, you see that that subset has a performance about 87, 80 plus, 7 plus, about 87%. Okay, and this now allows users to choose from the strength of the prediction the most reliable predictions and get an idea for how many of the residues that have an effect. If I want to find all the effect variants in my data set, how down do I have to go, or what fraction do I get when I look at only, say, reliability index 6, 7, 8. So I assume that I will find less than 5%. So what I have typically is less than 5% of all the variants, okay? But for those, I expect that the performance here is above 96%. Okay, now what you see here are two curves. One red for the effect, one green for the neutrals. They have different shapes. We go into the next phase where now I'm going to show you, again on the same axis that I had before, where minus 100 is very strongly predicted to be neutral, plus 100 is very strongly predicted to be effect, and I'm going to open the can of the binary classification. So far all I had is neutral versus effect. Now I'm going to introduce something in between. So I'm going to have strong effect, strongly neutral experimentally, and intermediate. And I'm going to ask my system that was only trained on my 2 effect, So the same as I had here, simply if I had a different experimental label, how would the system look? And the answer is that the ones with a stronger effect are more on the right side. The ones that are stronger neutral are more on the left side. Blue, the ones that have sort of an intermediate effect are sort of undecided. They are more in the middle. Now what that means is that the system actually takes apart strong effect and weak effect. So the system, the strength of the prediction is not only reflective of performance, as this one shows, the stronger, the better performance, but it also distinguishes the strength of the effect. These things are related, they are not that surprising. We may argue, and that is back to what Natalie said before, we may argue that the reason, in fact, why some samples are presented more often, why the signal is clearer for some, is because the signal is stronger. The effect is so strong that all the signals, all the samples in that input space point into this direction of change, and strongly so, all right? This is why this is not surprising, but it is actually not evident. Now, there are some examples, let's just go over them, and I don't want to go for the details here. Now we can sort of map this prediction onto a three-dimensional structure, and now we can go, and I will get more into that on Thursday. Now we can actually, since we have a method that predicts a variant for every, or that predicts an effect for every single variant, I can now take this method and predict for all 19 non-native amino acid changes that are possible there, what would be the effect? And that is ultimately what you sort of see here. Yeah, I'm sorry, a little bit, it's an old slide, quality is getting a little bit better. So what you see here, or what you could possibly see, is the darker the red, the more all 19 non-native are predicted to have an effect. The more blue, the more the 19 non-native are predicted to be neutral. If I rotated this, what you could see then, no, it's not a movie. So maybe on Thursday, the movie is still in, where it rotates and you see it a little bit better, at the expense that here the red is better than in the movie. But in the movie you see a little bit better that essentially everything around the binding site here is red, and a little bit of that is true in this other binding site. So you essentially see that where you have the strongest effect in terms of 19 non-natives, so for all possible variants, that is where the binding site sits. And that is what we are going to do again on Thursday. Now you can compare the performance to other methods. Here, the method SNAP in a Q2 two-state accuracy versus Polyphen, sift, and random. You immediately see that sift and Polyphen, essentially simple optimizations that take an afternoon to, well, Polyphen takes an afternoon, sift takes an afternoon, Polyphen takes much more than that, but it's still relatively simple. This took many years to do, but they both improve over random much more than SNAP improves over them. Okay, that is one simple reality. Now, there are other scores you could look at, accuracy, precision, recall, and whatever these are called, two-state accuracy, there are different scores. And let's go back to this one here. Now you can argue, okay, we have three different prediction methods. Whatever residue they predict the same way is a simple classification task. Any method can do it, okay? Let's just take all of these out, the simple residues. Let's only look at the residues where at least one of them disagrees. Let's put them as 100% of my data set and reassess how good are the methods on those not simple methods. So let's call them not simple variants. Let's call all the ones where everybody gets the same answer as simple ones. Let's cut them out and let's look at the non-simple ones. For the non-simple ones, SNAP looks like this. So this goes clearly down from this slide here, you see, substantially. But now you see that sift and Polyphen go even further. So what sift and Polyphen capture are to mostly, and also to a large extent what SNAP captures, are sort of relatively simple features. What SNAP adds that is relevant here through machine learning is something that for all the remaining things, it still does way better than random, while they no longer do this. Yeah?
How can that be worse than random?
Yeah, so that is, we have to be careful. This is ultimately because we slice the pie in a different way. When you look at sift, let's just focus on sift, okay? When you look at sift, sift is way better than random. Now, when you remove the ones where they all agree, essentially you remove most of the ones where sift is right. For random, you don't do that. Because for random, you have a random probability of predicting. So for every new data set, whether it's a difficult one or not, you get the same random number. Because this is a random number generator, right? So the random is not a biological random. It's a computed random. So this number will not change. While this number will change because you essentially remove whatever it works for. The ones that they all have in common are the ones that are simple, that are the ones that work. And if you remove everything that works, you get something that is less than random. Okay, that is somehow the flip side of the coin. That is the way it can be. So it's completely counterintuitive. Now, you cannot just take that and say, okay, for the remainder here, I'm going to take a coin instead of sift. Because you don't know which ones they are, right? Well, actually you do. Actually, you could do that.
You could even say...
you could actually do that.
Sift predict, I take the other way around.
That's actually a very good idea. Yeah, you're fully right. It's true. You're 100% right. It's true.
It's just in that one data set. Maybe it's very different than another data set. Why should it always be worse than 50%?
Well, because essentially you take away everything where it works, right?
Yeah, for the others it might also work.
No, but that random is not the others. Yeah, but this one, so the Nabil idea works for the others as well, yes. It just doesn't, for Snap it would be bad. So your idea would be optimally bad for our method. But would be good for poly. I will show you, let's bring that back in a moment. Max Hecht worked on Snap 2. In Snap 2, there are a couple of things that he wanted to add. One is bring in disease variants. One is bring more neutral from EC, so getting an overall of 100, 101, so 20% increase of the overall number. Add some particular prediction features, such as co-evolution or contacts, evolutionary information, biophysical features in a way that we had not used before, some annotations in a way that we had not used before. So the overall number of features has increased. So another thing that has been added here is disordered regions, contact potentials, correlated mutations, low complexity. Those have all been added, and some particular features, the binding site features. But the most important change ultimately, so retrain larger data set, but then also have different versions. So I have a version that doesn't use alignment information and a version that doesn't use the disease data. Because then you can sort of differentially see whether a particular variant, if the disease, so you get an effect. To predict an effect of a particular variant, and then you take the version that doesn't use disease trained, then, and you still get an effect, and you assume this is a molecular effect. Okay? If you see that suddenly, wait a minute, if with and without disease it gives an effect, and with the molecular it doesn't give an effect, then you assume it's a level organism. It's not so much on a molecular level. Anyway, on a differential way, you can somehow get some rationale. The alignment story the same. If you see an effect, although you don't use the alignment, then it's not about conservation. It's about change in secondary structure or something like that, right? You get these curves again that I showed you before. Again, the new method is slightly higher. Let's just keep it there. From neutral effect, it's the same thing. And now we are back to the story that I said before. The not so easy to do. Snap 2, Snap, and this is a different data set, and now this is actually Polyphen 2. And for this different data set, so Daniel is in this particular case totally right, but in this particular case, sift actually is slightly better than random. So Nabil suggested in this particular case, for this particular data set, would do slightly worse, but inverting it really would sort of fall here, right? But within the error, it wouldn't matter. For Polyphen the Polyphen 2 is now much better for these, but again, in fact, even better than Snap. The first version of Snap was, but this one is still doing better. You have a reliability index, as I showed you before. Now the curves are no longer as linear as they were before. You can even do that for residues without alignments.